What you are looking at in this droplet is the final result of a precise laboratory workflow, pure recombinant human insulin. Over the next few minutes, we will manufacture this protein entirely from scratch. The strategy requires two main components: human genetics and bacterial hardware. We will isolate the specific human gene responsible for producing insulin, stitch it into a circular DNA vehicle called a plasmid, and insert that plasmid into E. coli. The bacteria will then act as microscopic manufacturing plants, producing human insulin on demand. This interface displays the specific biological and chemical reagents required for the protocol. We need trisol for extraction, the PET28A plus vector to hold the gene, restriction enzymes to cut the DNA, competent bacterial cells to host it, and NIC-NTA resin to capture the final protein. This process involves dozens of precise chemical reactions. By organizing the workflow into 4 verifiable phases: gene preparation, plasmid construction, host transformation, and protein purification, we maintain strict control over every variable on the microscopic assembly line. Phase 1 begins with extracting RNA from human pancreatic tissue using a chemical lysis reagent called trisol. Before proceeding, we measure the purity of the sample using a spectrophotometer. An A260 over A280 ratio between 1.8 and 2.0 confirms the RNA is clean enough to use. Raw human RNA contains non-coding segments called introns. Because bacteria cannot process introns efficiently, we have to convert our RNA into a streamlined format called complementary DNA, or cDNA. This schematic shows exactly how that conversion works. We introduce an enzyme called reverse transcriptase, which reads the original RNA sequence and synthesizes a stable, continuous cDNA strand containing only the active instructions for making insulin. Once formed, we amplify the insulin gene using a polymerase chain reaction. Engineered primers attach to the cDNA and rapidly duplicate it. These primers add 2 structural anchors to the sequence ends, cut sites known as EcoRI and HIND3. This glowing band on the agarose gel confirms the PCR was successful. By running the sample under an electric current, the DNA separates by size. A distinct mark exactly at the 330 base pair length proves we have accurately amplified the human insulin gene. We cut this band directly out of the gel and dissolve it in buffer to extract the DNA. The insulin insert is now highly concentrated, purified, and ready for integration. Phase 2 shifts to the delivery vehicle, the PET28A plus plasmid. This is a robust circular DNA molecule that naturally replicates inside bacteria. To insert our insulin gene, we first have to physically open this closed loop. In this diagram, restriction enzymes are acting as molecular scissors. We use the exact same enzymes, EcoRI and HIND3, to digest both the plasmid and the human insulin gene. This creates complementary, overhanging sticky ends on both pieces of DNA. To verify the plasmid is fully opened, we run a small sample of the digested mixture alongside an undigested control plasmid. If the digestion is complete, the cut plasmid will travel differently through the gel, confirming the circle is broken. Next, we mix the cut plasmid and the insulin insert at a precise 3:1 molar ratio. We add an enzyme called T4-DNA ligase. The complementary sticky ends find each other, and the ligase permanently fuses the backbones together. The resulting structure is a single continuous loop of recombinant DNA. The human sequence is now seamlessly integrated into the bacterial delivery system. This microscopic view captures the physical insertion step, called heat shock. We mix our recombinant plasmid with competent E. coli cells on ice. We subject them to 42 degrees Celsius. The sudden temperature spike compromises the bacterial membrane, forcing the cells to absorb the surrounding plasmid. We return the cells to ice to close the membranes, add a nutrient-rich SOC medium, and let them recover at 37 degrees for 1 hour. Then we spread the liquid culture over agar plates containing the antibiotic kanamycin. On this petri dish, the results of the antibiotic selection are visible. Our PET28A plus plasmid carries a specialized gene for kanamycin resistance. Therefore, any bacterial colonies that successfully grow on this plate must contain our plasmid. If the plate is empty, it usually indicates a poor ligation. If colonies grow but contain the wrong sequence, the initial restriction digestion was likely incomplete. This chromatogram displays the final quality control check. We isolate the plasmid from a successful colony and send it for Sanger sequencing. Sharp, distinct peaks confirm that the insulin gene is perfectly aligned and entirely free of mutations. With the sequence perfectly matched to the human code, the bacteria is fully reprogrammed. The engineering phase is complete and mass manufacturing begins. We extract the verified plasmid and transfer it into BL21, a highly specialized strain of E. coli engineered specifically to express or produce massive amounts of foreign protein. This comparison shows the exponential growth phase. We incubate the BL21 culture in a broth until it turns cloudy. We measure this density with a spectrometer. We must wait until the culture reaches an exact optical density, or OD600 metric, of 0.6 before triggering production. At 0.6, the bacterial population is perfectly primed. We add a chemical inducer called IPTG. This chemical activates the specific promoter sequence on our plasmid, signaling the bacteria to halt normal functions and start aggressively manufacturing the human insulin protein. Temperature control during this phase is critical. If the incubation temperature is too high, the rapid production causes the insulin to misfold into dense, insoluble clumps called inclusion bodies, rendering the protein completely useless. Looking at this column cross-section, you can see the final extraction. We break open the bacteria and pour the entire cellular mixture through Ni-NTA resin. Because our plasmid added a chemical His tag to the insulin magnetically binds to the resin. The rest of the cellular waste washes straight out. We then wash the column with an imidazole solution, which safely releases the bound insulin from the resin. The biological machinery has finished its job. To confirm that the liquid we just collected is actually the protein we intended to build, we perform one final analytical test, an SDS-PH gel. This thick, isolated vertical band on the stained gel provides definitive proof. The location of the band matches the exact molecular weight of our insulin fusion protein, and the lack of other marks proves the sample is highly purified. The exact workflow we use to isolate, amplify, insert, and express this insulin is a universal foundation. By swapping the insulin sequence for other specific genetic codes, researchers use this exact sequence of techniques to produce therapeutic nanobodies, engineer viral proteins for vaccines, and synthesize vectors for gene therapy. By treating DNA as a programmable code and the bacterial cell as a localized foundry, we gain the ability to accurately manufacture the complex molecular structures that sustain human life.